Hej och välkomna tillbaka till Erik Penser Banks Healthcare-dag 2023. Jag heter Claes Palin och är aktieanalytiker här på banken. Vi ska gå vidare i vårt spännande program. Och nästa bolag som ska presentera är Spago Nanomedical. Och för att presentera Spago har vi bolagets vd Mats. Välkommen hit.
Tack så mycket.
Varsågod.
Tack för det. Jag kommer att köra den här presentationen på engelska. And I start to say welcome to this presentation and update of Spago Nanomedical and our projects in precision imaging and radionuclide therapy. Despite major progress in the fields of medical imaging and cancer diagnostics, there is significant remaining medical need for improved diagnostic precision and treatment effect in many patients. At Spago Nanomedical, we have developed a platform, a unique platform for physiological targeting to tumors. This is based on a phenomenon called the enhanced permeability and retention effect, or the EPR effect, that essentially derives from the disorganized and leaky vasculature surrounding many solid tumors, and that allows diffusion of molecules of certain sizes and compositions into the tumor tissue. The EPR effect can be exploited for delivery of precisely designed molecules to tumors. What this means more specifically is that physiological targeting mechanisms allow for precision delivery of functional nanoparticles to tumors. Based on this technology platform, we have designed functional nanoparticles for use in both imaging and radionuclide therapy. We focus on two main development programs, SpagoPix and Tumorad. In SpagoPix, we developed a manganese-based MRI agent for precision imaging, which provides clinical validation to the platform, as we shall see later in the presentation. And with Tumorad, we developed a new radionuclide therapy based on the clinically validated isotope lutetium-177. The candidate drug Lu-177SN21 is now well on the way into clinical trials. Functional nanoparticles are, there's the Tumorad. Functional nanoparticles are different from traditional nanomedicine in the way that they carry inherent capacity to perform as imaging or therapeutic agents by their sheer presence within the tumors or the target lesions. And now you wonder, does this work in humans? Well, yes, it does. We have positive clinical results in breast cancer with SpagoPix that confirms selective accumulation in human tumors of our functional nanoparticles. And imaging with Tumorad also shows promising tumor distribution in preclinical models. This indicates we have a robust technology for physiologically targeted precision medicine, with consistently good accumulation seen both in animals and in human tumors, and this opens up for use both in imaging diagnostics and in therapy. So following this major breakthrough, we continue to pursue our projects in clinical development. We aim to position our projects in indications of high medical need, and especially in devastating diseases mainly affecting women like breast cancer, endometriosis, and ovarian cancer. So now let's have a closer look at our projects, and we'll start with SpagoPix. MRI is commonly used because it has a very high spatial resolution and sensitivity. MRI cameras or machines are readily available in many places today. And the use of medical imaging and MRI in particular is expected to increase by the increasing use of screening tests and because the fact that MRI does not incur harmful radiation to the patients. So MRI is supposed to be more common. Well, The specificity of MRI is, however, not always as good as you would expect or could hope. And this lack of specificity sometimes restrict the use of the images and necessitate various different follow-up procedures, which is suboptimal and costly. So, as SN132D selectively accumulates in the tumors, this allows us to bring increased precision to MRI and to the patients in indications where there is a clear need for improved medical imaging. We already see good tumor selective contrast with the SpagoPix product candidate SN132D in patients with breast cancer. The tumor selectivity and lack of background enhancement provides potential for better imaging precision, which in turn could lead to better outcome for the patients. This could be in surgery or in actual raising or setting the diagnosis for the patients. More importantly, or in addition, I should say, these images provide clinical validation of the physiological targeting mechanism for our platform material. And this not only supports continued development for SpagoPix, but it also provides support for clinical translation of our Tumorad radionuclide therapy in different solid tumor indications. The things that make SpagoPix and SN132D stand out in comparison to contemporary current contrast agents for MRI is the fact that it is selective for malignant lesions. And as we shall see later, also for certain other types of lesions. This is based on the physiological targeting that I explained previously. In turn, this creates a potential to reduce false positive imaging findings, which is a problem today, especially in breast cancer. And the reason for that is that the background enhancement in the images is more or less non-existent. One other interesting thing about this is that this lack of background could actually support machine reading of the images. AI, for instance, could be used in a more effective way to set the diagnosis in these images. We also have the potential to reduce the MRI scanning time, as this agent can be infused outside of the MR suite, which makes the actual imaging capture more effective. It also allows for more flexible imaging, as the agent stays in the body for up to 4 or 5 hours, which prolongs the window from today's about 5-10 minutes quite substantially. And finally, this agent is manganese-based. It's not based on gadolinium, which is used in more or less all other MRI contrast agents today. With this in mind, and as a way to further position SpagoPix in different target indications of high medical need, we see good opportunity not only in cancer, but also in endometriosis. The physiology of endometriotic lesions is very similar to cancer in some key aspects, and this supports physiological targeting in the same way as we've seen with tumors. As we already demonstrated the principle for targeting in breast cancer, we now set out to explore if we could improve and shorten the time to diagnosis in this devastating disease where the average delay to diagnosis is very long. There is also a great need for endometriotic patients to improve the imaging precision in order to support surgical intervention and treatment. Endometriosis is underserved. It's a devastating disease that affects up to 10% of women of fertile age. It has huge personal and societal consequences. if we can show that SN132D actually works well in this patient group, the potential market could be substantial. I will now switch project to Tumorad, and we'll start with some basic facts. As radioactivity has been known for a long time to be effective in the treatment of cancer, and radionuclide therapy is a way to bring radioactivity to the tumors within the body. Radionuclide therapy has been used historically and successfully to treat thyroid cancer, lymphomas, and to relieve pain from skeletal metastases. In the last 5 years or so, we have seen significantly increased interest and investments in this field as new generations of radionuclide therapies for the treatment of neuroendocrine and prostate tumors has entered the market. This has encouraged a growing pipeline of new radionuclide therapies, but most of them come with the limitations that they are restricted to the tumor type they can actually go for and address by means of their targeting mechanism. So essentially one agent means you can target one indication. Now what if we can provide one agent to address major needs in several cancer indications? Let's see how this could work. We already see or have shown that we have clinical science and clinical proof that our functional nanoparticles do accumulate selectively in human tumors. Further support was recently published preclinical data that shows that we also have good accumulation on par with or even better than the approved benchmark radionuclide therapy, Lutathera. We see favorable distribution and very good radiostability of the candidate drug, and we also see that it inhibits tumor growth and prolongs survival in preclinical models of aggressive cancer. And with these results, we feel confident to take Tumorad into clinical trials, into humans, and we aim to start the first phase 1/2A trial within very shortly. In order to put Tumorad into some kind of market context, let's have a look at the current radionuclide therapy field. This can be roughly described in 2 dimensions based on type of radiation and type of targeting. So biological targeting means that you can essentially target one tumor type or at least a restricted number of tumor types, whereas physiological targeting, as we've seen, have broader utility. And one group of agents use short-range but high-energy alpha radiation. Among these we see Spago, which has been approved since 2013 for skeletal lesions from prostate cancer. Another larger group of agents use beta radiation. This is the most common type of radiation used for radionuclide therapy. Among these, we also find the newer agents, Lutathera and Pluvicto, that are both marketed by Novartis. And both of them also use, by the way, lutetium-177, the same isotope as we use with Tumorad. And then we have Tumorad. And as you can see, there are not many agents in the pipeline that combines beta radiation with physiological targeting. And as we already know, lutetium is a clinically effective isotope, and that physiological targeting works with our particles. Well, this could allow one radionuclide treatment for many different cancer tumors. So if we look even closer to what this uniqueness could mean, we can conclude that it provides potential for a strong competitive position and use in several different indications. And listed here are some tumor types that we know have an EPR effect that is clinically documented. As you can see, it includes some large indications and some smaller indications. But even if we use mortality rates as a rough proxy for the total population size that could theoretically be addressed, we get to enormous addressable market figures. Also, if we use conservative estimates, the total theoretical market is substantial. But of course, we will not target all of this at once. What we are looking for and are very interested in to explore further is ovarian cancer, an indication where there is not so many treatments approved and where there is a huge medical need. There is also the opportunity to go for an orphan indication, an orphan drug development strategy in this indication. So, to conclude, I believe that Spago Nanomedical is well set to progress our platform project further into clinical trials and to further clinical milestones, and eventually address major clinical needs, both in cancer and in endometriosis. We know that we have clinical validation for the platform technology by means of SpagoPix imaging in breast cancer. We have very good support, preclinical support for Tumorad for it to enter clinical trials, and that is what we are doing right now. We have a phase 1/2A in preparation. The study will start in Australia, and more news will come on that very soon. We have an established CMC process and logistics for GMP production, which is not to be forgotten. And there are also the options for imaging biomarkers for use with Tumorad if we wanted to select patients based on availability or presence, rather, of the EPR effect in the tumors of the patients. So with this, I thank you very much for your attention and would be delighted to take questions.
Thank you so much, Mats, for a very exciting presentation. And let's start with SpagoPix then. And the EPR effect is a very interesting one. But how well documented is that in endometriosis?
Well, there is scientific support, and as I mentioned, the physiology of the endometriotic lesions share common key features of tumors, like leaky vasculature, inflammation, those types of things that tells us that physiological targeting by our particles could work in that indication.
And choosing between endometriosis and breast cancer, do you believe this is a better opportunity, or what is the reason why you are doing this clinical study?
We believe it's a huge indication with a huge medical need, and we also see it as a way to, with rather limited resources, take the project one step further into phase 2. And position it as a way to use MRI in major women's health indications. Okay.
And in endometriosis, you mentioned that there's quite a long time before diagnosis often. But how is the current diagnostic standards within the indication?
Mainly ultrasound, sometimes laparoscopy, that is keyhole surgery, is also used, which has a poor precision. MRI without contrast is sometimes used. It's not recommended, but it's still used sometimes. All of this has very, very poor precision, which kind of leads to the very long delay in correct diagnosis for these patients.
Do you believe it could be a challenge to enter an indication where you don't have a recommended standard that you are pursuing?
Well, I mean, this is a way for us to position the project in a big indication. We believe that as MRI is used anyway, it's readily available in the clinics, and it's much better than ultrasound in the way that it provides better resolution, it provides more information for the surgeon who is supposedly going to take these lesions away. I think that the need is there, the infrastructure is there, and the willingness, I would say, among these patients and their treating physicians is definitely there.
Mm-hmm. And the clinical trial is up and running. The first patient has been dosed, I guess is the right name. And maybe you could help us out. The recruitment rate is according to plan?
Much faster than in breast cancer, at least. No, it goes very well. We expect to have this trial completed by the half year or something like that. Mm-hmm. So around summertime.
Okay, perfect. And that would be top line data?
Yes.
Okay, perfect. And if this data package looks promising as you expect, what would be the next step from your side?
Well, first of all, I should be clear about that. This is kind of a way for us to try Spago Pix in a new indication. So it's difficult to know exactly what to expect. But if it works, then it's great. Sorry, what was your question again?
What would be the next step?
The next step, yeah, sure. I mean, again, we are positioning this in a way that hopefully makes it attractive for partners. But if not, we believe that the next step could be rather limited in terms of size to be able to reach registration. So we are looking at different alternatives as we speak. Of course, with being very cautious about the budgets.
That's good. Sounds very good. And then jump to Temadag. You are progressing towards the start clinical study. What's happening right now with the program?
Well, we are going to send in the application to start the trial very soon. I'd hoped it'd be today actually, so it's any day now. We are going to start in Australia, also as a way to speed up time to clinic, as the registration or the application procedure is different there. It's faster than in Europe and in the US, for instance. And also, since we set up an Australian daughter company in order to draw from the tax credit that Australian government gives to R&D activities.
And that is up and running now?
It's up and running now, yes.
Okay, and you don't see challenges to run a trial on the opposite side of the globe?
Not at all. We have good people on the ground there. We've set up quite an optimized organization with the CRO, with certain project management related tasks on other consultant and radio labeling facilities as another key one. So no, it seems to work very well so far.
And how much would you say to run a trial in Australia compared to in Europe?
Well, the tax refund for R&D is around 43%, so it's quite substantial. Okay,
sounds great. And if you just could remind us about the study design.
Yeah, so what we are looking for is to start the phase 1 part where we do dose escalation in an optimized way to get us to a clinically effective dose, hopefully quite quickly. And also to provide initial dosimetry, telling us the proportion of how much drug goes to the tumor and to other parts of the body, since that balance is critical for a drug like Temadag. That is designed to include up to 30 patients. We do not believe that we will need 30 patients, but that's the design. The second part is a phase 2A, where we intend to go into discrete populations of cancer patients. For instance, ovarian could be one, triple-negative breast cancer could be another one, where we see clear need for this type of treatment.
And as you mentioned, ovarian cancer is something that seems to be on your priority. But why is ovarian cancer such an interesting indication for you?
Again, the medical need is high. There are a few kind of lines of therapy approved. Those lines are interesting also in the way that they could potentially benefit from a radioactive drug in combination. We see, of course, as I mentioned, the potential to go for an orphan strategy in that indication as well. So there are several things that makes us interested in that.
And the EPR effect is well documented?
It's documented, yes.
Okay, perfect. And as you showed during your presentation, physiological targeting, you're quite alone, it seems, at least in the radionuclide field. Why do you think you are alone so far?
I would correct that a bit. We are not alone when it comes to the traditional nanomedicine, which has used the EPR effect of physiological targeting for a long time as a means to deliver things into the tumors. I think that the reason we are alone in this field is that it's rather complicated to design a material that can withstand the radiation emitted from the isotopes. So getting to that stability of the molecule is not an easy task. The other thing is that I think it's been more kind of easily conceptualized to kind of tag an already existing antibody for a certain target with the radioisotope, rather than embarking on this kind of more complex material that we have. I mean, the reason we are doing this is that we already have this material, since it's been developed for a completely other project, the MRI contrast agent. So this is a way for us to build on the platform we already have, which is known now to accumulate in tumors.
And what would be the biggest opportunity to use this physiological targeting compared to biological targeting?
Well, it is certainly it provides a means to get to tumors that you can't with existing antibodies, where there are no clear biological signals to target to. So we know that the EPR effect is present in many solid tumors and metastasis, especially fast-growing tumors and metastasis. So it provides a way to differently attack the tumors.
In this clinical trial that is about to start, what would be the biggest challenge to use this targeting and with a radioactive drug, do you believe?
Well, as in all, it's difficult to say. I mean, we are going to do a phase 1 first to show that it's safe. And safety in this case will mean radiation safety.
Yeah.
And the Temadag project in itself is all about the balance, getting drug to the tumors and trying to avoid getting it to other places in the body. So that will be the main challenge.
And my last question then would be, what should we expect from Spago this year when it comes to the news flow?
Yeah, well, first of all, as I said, the start of the Temadag clinical trial, which we expect to happen also before the summer. Then we have the results from the endometriosis phase 2 trial, which is also expected to come before summer. So there are some big things already this first half of the year. And then, of course, we will have initial data from Temadag, dosimetry, biodistribution, PK, things like that can tell us if Temadag has what it takes to get up to therapeutic doses in different types of tumors.
And that we will have in 2023?
We expect to have that, yes.
Okay. Sounds exciting. Thank you so much.
Thank you.
Thank you.